Here with me I have Ramesh, who works for Fluidigm. And if you would, first of all, Ramesh, tell me what you do for the company.
Sure. I'm the director of molecular biology over here. That's sort of a very vague term. But what essentially it does is, since Fluidigm is based on using a number of molecular techniques for detecting various elements associated with genetic diversity, my group and I typically work on developing applications where these kind of techniques can be used. One of the techniques which we apply this kind of approach for is for the field of genotyping. And genotyping includes a number of different approaches.
Well, and let's maybe go over a couple of quick definitions for people who may not understand even what genotyping is, but I know we're talking about SNP genotyping as well as microsats, for example. What are those?
Okay, let's back up over here first. Genotyping typically means trying to determine what the particular genes are which is associated with either an individual or a species or a bunch of different individuals, genes which are common between different species, etc. That's just what genotyping means, the type of gene is good. There are a number of different variations on the genome among different individuals. One of the most common ones is something called SNPs, and SNPs refers to single nucleotide polymorphisms. Let's back up a little bit further over here. Genes are typically made up of what we call nucleotides. It's a very, very simple, compact code which defines what kind of genes are present in an individual, what kind of proteins are expressed, what kind of characteristics each individual has. Genes are made up of, as I said, of a number of nucleotides. A change in a nucleotide sequence or sequence of events is called a polymorphism. So a SNP is a single nucleotide polymorphism, which means that an individual can be different from another individual by just a single nucleotide change. That's what a single nucleotide polymorphism is, or a SNP. And that's actually a very valuable marker for looking at individual differences between species, individuals, etc. There are other types of polymorphisms in the genome. One thing which is found in a number of different genes and genomes is we find what are called repeat sequences. So there are typically 4 nucleotides, A, C, G, and T, which are expressed in a number of different combinations. Let's say, for example, you have a repeat. Okay, let's say AG. Okay. So in other words, you see AG, 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 AG. That would be called a dinucleotide repeat. A microsatellite essentially is a number of such repeats. You can get trinucleotide repeats, in which case it might be AGA, AGA, AGA. But the way microsatellites are used is each individual will have a number of different microsatellites which are composed of these kind of repeats, a certain number of repeats. So this is really useful, for example, if you're trying to distinguish one individual from another. Did that answer your question?
Yeah, I think that helps give us a little bit of background, because the next, let's look at agricultural biotech, for example. How would this kind of genotyping be used in agricultural biotechnology today?
Okay, so let's talk about why genotyping is actually important at all in agbio. It's really important to be able to track whether a plant is what we think it is, for example. Let's say, for example, we've got a plant which is resistant to a certain type of pesticide. We really want to make sure that the stuff which we are planting actually corresponds to that. One thing which is very characteristic of the agbio business is that seeds, high quality seeds, are worth their weight in gold. And that's not an exaggeration, it is actually true. So planters as well as sellers want to be absolutely certain that the seeds that they are selling or buying is exactly what they expect it to be. So that's exactly why they want to look at they're not really interested in looking at differences. They want to make sure that what they have is exactly what it is. In other words, genotype it accurately. Traditionally, what people have done is used microsatellite detection. A couple of reasons for that. One is that it's a much older technique. It's been around for quite a while, at least 20 to 30 years. It is relatively easy to perform once you've been trained. And it requires, the running cost is relatively low. What's the disadvantage of that approach? The disadvantage is that because it's based on technology which is 20 or 30 years, the world has moved on. There are simpler ways to get the same answer. The need of the hour is not just low running costs, but it's actually turnaround time. And microsatellite detection, because the way it was configured, was meant to be hands-on, laborious, and not automation friendly, not amenable for high throughput screening, for example, which is why people have been in the process of switching over to SNP genotyping in order to characterize their different plants.
So, Ramesh, when it comes to SNP genotyping and being used in agricultural biotech,
Sure.
what role does Fluidigm's product or system play in that?
It's a really good question. So one of the advantages about the Fluidigm system is that it's extremely flexible. Typically, when you're looking at other genotyping applications or products which are out there, they are extremely tailor-made for a particular type of assay, a particular type of sample. It just doesn't lend itself for much flexibility. So the Fluidigm platform is actually extremely flexible, which means that any sample can be tossed in. It doesn't necessarily have to be of any particular genre. The second thing is, as you know, one of our core technologies is the fact that we're able to make chips which run extremely small volume reactions. What that does is two things. One is that it reduces the overall cost, and this is actually a really important point as far as the ag bio business is concerned, because since they deal with so many thousands of samples, so many thousands of different genes, they are extremely cost sensitive. So cost per data point is the way they look at it. And so this particular aspect of it is really important for them. Again, because of the reduced volume and because of the fact that we have an integrated system, our time to result is really fast. It's really short. And additionally, the fact that our system is completely automated means that there is no ambiguity related to the results. So one of the perceived advantages of microsatellite detection compared to SNP genotyping, which champions of the microsatellite technology have always spoken about, is the fact that microsatellite detection methods are unambiguous. Anyone can look at the result, they'll know exactly what it says. SNP genotyping is more subjective. That's not the case when you use the Fluidigm technology. I mean, they are very, very clear and unambiguous results which you get. So that actually takes care of a lot of the uncertainty around looking at specific genotypes associated with specific plants.
Well, it sounds like the Fluidigm system really does provide an advantage for companies that are doing research that would be, whether it's SNP genotyping or microsat for that matter. Well, thank you very much, Ramesh, for bringing us up to date and explaining some of how the Fluidigm system can be applied in ag biotechnology. Here at Fluidigm headquarters, I'm Chuck Zimmerman reporting.